Hey everyone, and welcome to another deep dive. You know how you're interested in cell culturing. Well, today we're going to explore that fascinating world of growing cells outside their natural environment.
Like in a lab.
Exactly.
Yeah.
And get this, our source material is 2 YouTube videos from Mohammed Imran Sajid. They're like a virtual tour of a cell culture lab.
It's pretty amazing what scientists can do these days, taking tiny microscopic cells and basically recreating the perfect conditions for them to grow in a lab.
I know, right?
Yeah.
It's like creating this little customized world just for these cells. But, you know, when I think about cell culturing, I kind of picture scientists in like hazmat suits working in these super high-tech labs.
Well, I mean, there is a lot of precision involved and specialized equipment for sure.
Yeah.
But it's not as intimidating as you might think.
Okay, good.
So let's break it down.
Okay, let's break it down.
Starting with the basic setup.
Right, the basic setup, lead the way.
Okay, so first things first, sterility is key.
Okay.
We need a very controlled environment to prevent contamination.
Yeah.
So think of it like, imagine a surgical operating room, but for cells.
Okay.
So they use these specialized workspaces called biosafety cabinets,
Oh, okay.
where filtered air keeps like those pesky microbes out.
Gotcha. So it's kind of like a mini clean room,
Exactly.
but for cells. All right, and then what kind of tools are we talking about here?
So you can kind of think of it like a well-equipped miniature kitchen.
Oh, okay. Interesting.
You've got your pipettes.
Pipettes.
Which are like tiny, super precise measuring cups and droppers for transferring liquids.
Oh, okay.
Then you've got things like falcon tubes,
Okay.
which are essentially little storage containers for holding those cell samples.
Okay.
And of course, you need ways to keep track of those cells, so that's where cell counting slides come in.
Okay.
It's a very organized operation.
Very organized. It sounds like it. What about those special dishes or plates? I feel like I've heard about those.
Ah, yes, the cell culture plates.
Yes.
They're like miniature muffin tins,
Right.
but instead of batter, we're adding cells.
Okay.
And just like muffin tins come in different sizes,
Right.
so do these plates.
Yeah.
We'll be exploring 24-well, 96-well, and 6-well plates,
Okay.
each with its own purpose depending on the experiment.
Gotcha. All right, so we've got our sterile workspace, we've got our tools, we've got our cell condos all prepped.
Right.
So where do the actual cells come in?
That's where it gets really interesting.
Okay.
So the cells in these videos, they start their journey in flasks,
Okay.
but they need to be woken up, so to speak, and prepped for further action.
Okay, how do we coax those sleepy cells out of their cozy flask?
So scientists have a more elegant solution than singing them a lullaby.
Okay, good.
It's a special enzyme called trypsin.
Trypsin.
Yeah, and it's like, it kind of acts like a gentle release agent. It helps detach the cells from the surface of the flask.
So it's almost like, I don't know, like when you add baking soda to a cake pan to try to get like that stubborn stuff out.
Yeah, perfect analogy.
Like loosen things up.
Yeah,
I imagine you can't just like dump a bunch of trypsin in there, right?
you're exactly right. Precision is key here.
Okay.
So just the right amount of trypsin incubated for the right amount of time, which is usually about like 3 to 5 minutes.
Okay.
Too little and the cells stay stuck. Too much and they could get damaged.
Gotcha. So it's all about finding that sweet spot.
Exactly.
So once the cells are detached, they're floating free. What happens next?
So we need to neutralize the trypsin.
Okay.
Kind of like adding an antidote. Fresh media full of nutrients for the cells does the trick.
Okay.
Then we give the flask a gentle swirl
Okay.
to dislodge any stubborn cells.
Okay.
Transfer everything to a new tube. Think of it like pouring those loosened cake batter remnants into a mixing bowl.
Okay, that makes sense. So now we've got a tube full of cells and media. What's next?
Time for a spin cycle.
Oh, spin cycle. Okay. But not the laundry kind.
Not the laundry kind.
Okay, good. All right. What's the cell spin cycle look like?
So we use a centrifuge.
Okay.
It's a machine that spins at high speed to separate the cells from the liquid media.
So it's like a salad spinner, but for cells.
Exactly.
It's like specialized tools for tiny tasks.
Yeah.
That's what I'm seeing. All right. What's the setting on the cell spinner?
So this particular spin cycle happens at 800 rotations per minute for 5 minutes.
Okay.
And that's enough force to send those denser cells to the bottom of the tube.
Okay.
They form a little pellet while the lighter liquid stays on top.
So it's kind of like separating the pulp from the juice.
Exactly.
And what happens to the liquid part?
We carefully remove that. That's called the supernatant.
The supernatant. Okay.
Leaving our precious cell pellet at the bottom.
Gotcha.
Now comes the crucial task of figuring out how many cells we have.
Okay, yeah.
You got to count it, right?
All right, we got to count them, but how do you count something so tiny?
Well, that's where a clever technique comes in.
Okay.
And a specialized machine, we use a special dye
Okay.
called trypan blue
Trypan blue.
and a biorad cell counter.
Okay, biorad cell counter. All right, break it down for me. How does this dye work?
Okay, so imagine trypan blue as a bouncer at a very exclusive club.
Okay.
It only lets in the cells that are, shall we say, no longer with us.
Okay.
So the dye can only enter dead cells,
Okay.
leaving the live ones untouched.
Oh, okay. That's smart.
This helps us easily distinguish between the living and the, well, not so living.
That's pretty ingenious.
Yeah.
So you mix the cells with the dye, and then this biorad machine does the counting for us.
Yeah.
No more squinting through a microscope.
Exactly. We load the mixture onto a special counting slide, insert it into the machine, let technology do its magic. It counts both live and dead cells, gives us a precise tally of our cellular workforce.
Okay. All right. So we got our cells counted. They're ready for action. What's next?
So next we have to seed them,
Feed them. Okay.
which means transferring a specific number of cells into those well plates that we talked about earlier. It's kind of like planting seeds in a garden.
I like that.
But instead of soil, we're using these specialized plates, and our seeds are tiny living cells.
I dig this gardening analogy.
Yeah.
So how do we know how many cells to plant in each well?
So it all depends on the type of plate and the specific experiment. Think of it like choosing the right pot size for your plant.
Okay.
So in the video, they used 3 different pot sizes, or well plates, 24 well, 96 well, and 6 well.
Okay.
And each requires a different, we'll call it planting density.
Okay. Different planting density.
So for the 24 well plate,
Mhm.
they use 50,000 cells per well,
Okay.
swimming in 400 microliters of media.
Okay.
The 96 well plate, you can think of that as like a mini apartment building, so it requires a lower density.
Okay.
5,000 cells per well
Okay.
in 100 microliters of media.
Okay.
And finally, the 6 well plate, our most spacious option. Right.
It needs 200,000 cells per well in 2 milliliters of media.
Whoa, okay, so many cells, so many calculations. How do we actually transfer those precise numbers into each tiny well?
It's a delicate dance
Okay,
of math and very careful pipetting.
okay.
We have to calculate the exact volume of cell suspension in media based on the desired cell count and the plate type. It's like a cellular recipe. We need the right ingredients in the right proportions.
It's not just about having a steady hand, it's about being a cell culturing mathematician as well.
Exactly.
What tools do we use for this intricate transfer?
We use a multi-channel pipette,
Okay,
which is like, a super efficient pipette that can handle multiple wells at once.
okay.
It's perfect for things like, adding 400 microliters of our cell media mixture to each well of a 24 well plate.
Okay.
So we've carefully seeded our cells into their new homes.
Okay, so we've seeded them.
Yeah.
What happens next? Do we just leave them alone for a while?
So we seal the plate
Seal it up.
to prevent any unwanted visitors.
Right. AAK contamination.
Okay, no contamination.
And we put them in an incubator,
Incubator.
which is like a climate-controlled spa for cells.
Okay,
It provides that perfect temperature and humidity for them to grow and divide.
awesome. So it's a waiting game now, right?
Right.
How long do those cells need to chill in their incubator spa before we check on them?
It depends on the cell type and the experiment, but it's usually at least a couple of days.
Okay.
In the meantime, we've got another crucial technique to explore. It's called subculturing.
Subculturing, okay.
It's like giving your cells a bigger house when their current one gets too crowded.
So it's like those microscopic cells are saying, hey, we need more legroom.
Exactly.
But how do we know when it's time to upgrade their accommodations?
So we have to recognize when the cells reach confluence, meaning they've formed a continuous layer,
Okay,
like a bustling little city covering the bottom of the flask.
I like that.
And that's their cue to say, time to move to a bigger place.
Gotcha. So we got a flask full of happy, thriving cells.
Mhm.
They're starting to feel a bit cramped. So how do we go about giving them that much needed upgrade?
So first step is we prepare their new digs.
Okay.
And this usually involves larger flasks,
Okay.
called T75 flasks. We add 15 milliliters of fresh media to each new flask, sort of like a welcome gift for our cellular movers.
Oh, I like that.
Yeah.
Making sure their new fridge is stocked.
Mm-hmm.
All right. So the new flasks are ready. How do we actually move the cells from their old, crowded digs to their spacious new homes?
So we carefully transfer a small amount of the cell suspension, about 250 microliters, from the original flask to each new flask.
Okay.
Think of it like taking a small cutting from a thriving plant and giving it its own pot to grow in.
Gotcha. So we've divided our cellular community and given them room to spread out. What's next?
Well, we have to keep an eye on our cellular tenants, right?
Right.
First, we label each flask so we can keep track of what's what.
Okay.
Then we make sure to change their media every 3 days,
Okay.
provide fresh nutrients to keep them happy and growing.
Happy and growing. That's what we want.
Yeah, and of course we monitor their progress carefully until they reach confluence again in their new homes.
So it's like being a landlord for microscopic tenants,
Exact.
providing the space, the amenities, and the regular upkeep to keep them thriving.
Right.
But how do we actually assess their well-being?
Yeah.
We can't exactly ask them how they're feeling, right?
Right, so that brings us to another fascinating tool
Okay.
in the cell culturing toolbox, the MTS assay.
The MTS assay.
And it's a clever way to assess cell viability,
Okay.
which basically means figuring out how many of those cells are alive and kicking.
MTS assay. Sounds maybe a little intimidating.
Don't worry, I'll break it down for you.
Okay, good. Bring it on. I'm ready to delve into the details. All right. So back to the MTS assay. It sounds a little intimidating, but I'm sure you can demystify it for me. How does this test actually work? Do we give these tiny cells a physical exam or something?
Not quite a physical exam, but close.
Yeah.
The MTS assay uses a special reagent. You can think of it kind of like a magic potion that reacts only with cells that are alive and metabolically active.
So it ignores the cells that are, let's say, taking an eternal nap.
Exactly. And here's the really cool part. This reaction, it produces a color change.
Okay.
The more vibrant the color, the more viable cells we have. It's like a built-in health indicator, a cellular mood ring, if you will.
The cellular mood ring. I love it. But I have a feeling scientists need more than just a visual cue to assess cell health. How do they get some hard data from this color change?
So that's where our trusty plate reader comes back into play.
Okay.
Remember how it measures light absorbance?
Yeah.
Well, in this case, the more intense the color, the less light passes through that solution. So the plate reader gives us a numerical reading
Okay.
that directly correlates with the number of healthy, thriving cells.
So high absorbance reading means we've got a party going on in those well plates. Lots of happy, healthy cells doing their thing.
You got it. And those numbers are gold for researchers.
Mm.
They can calculate the percentage of viable cells, compare results from different experiments. They can even plot graphs to see the impact of different treatments.
So they could see, for example, how a new drug candidate affects the health of cells,
Yep.
like compared to a control group.
Exactly. The MTS assay, it's a really powerful tool for studying drug efficacy and toxicity.
Yeah.
It can help researchers determine if a drug is actually killing target cells, like cancer cells, or if it's harming healthy cells as well.
It's amazing how much information we can get from a simple color change.
Mm-hmm.
But all this experimenting, it must create a lot of waste.
Mm-hmm.
What happens to those used plates and pipette tips? Do we just toss them in the recycling bin?
Definitely not.
Okay.
Remember, we're working with living cells here, and contamination is a constant concern. So all used materials, anything that's come into contact with those cells, is treated as biohazardous waste.
Okay, so no trips to the recycling center for those pipette tips. Where does this biohazardous waste end up?
It's carefully collected in these special biohazard bags and disposed of according to strict safety protocols.
Okay.
And that's not all. The entire work area, the biosafety cabinet, the surfaces, everything needs a thorough clean and with bleach solution to prevent any lingering contamination.
It's like a microscopic crime scene cleanup, but instead of dusting for fingerprints, we're like eradicating any traces of those tiny cells.
You could say that. Sterility is paramount in cell culturing.
Mm-hmm.
We need to protect those cells from contamination, and we need to protect ourselves as well.
Safety first. Speaking of safety, are there any risks associated with working with certain types of cells? I mean, some of these cell lines, they sound a little intense.
That's a great question. Not all cells are created equal in terms of safety.
Right.
Some cell lines, particularly those derived from tumors,
Okay.
can be more aggressive and potentially pose a greater risk to researchers.
So it's like handling a feisty house cat versus a gentle giant.
Exactly. So researchers, they need to be aware of the potential hazards
Right.
associated with the specific cells they're working with and take appropriate precautions.
Makes sense. Got to respect those cells, and they need to respect us.
Exactly.
Okay, so we've covered the ins and outs of working with cells, from prep in the lab and waking up the cells to seeding, subculturing, even cleaning up after the cellular party is over.
Right.
But let's step back for a moment and think about the bigger picture here. Why does all this matter? How does this microscopic world impact our everyday lives?
That's a crucial question.
Yeah.
You know, it's easy to get lost in those technical details and forget about the real-world impact of cell culturing. But trust me, this field is profoundly relevant to all of us. It touches our lives in countless ways, often without us even realizing it.
Okay, I'm intrigued. Give me some concrete examples. How does this world of tiny cells impact our big, messy human world?
Well, for starters, cell culturing is at the heart of so much medical research.
Okay.
Think about the drugs we take, the vaccines we rely on, the therapies that help us fight diseases.
Right.
Many of them were developed and tested using cell cultures.
So those tiny cells in those petri dishes are literally saving lives.
Absolutely. Cell cultures, they're playing a crucial role in the fight against cancer, Alzheimer's, HIV, infectious diseases. The list goes on and on.
Wow, I never realized the connection was so direct. It's easy to take those medical advancements for granted, but behind so many of them are teams of dedicated researchers working with cell cultures to unlock the secrets of human health.
Exactly. And it's not just about treating diseases, it's about preventing them as well. Remember Jonas Salk, the scientist who developed the polio vaccine?
Yeah.
He relied heavily on cell cultures to grow the polio virus needed for his research, paving the way for a vaccine that has saved millions of lives.
Wow. It's amazing to think that something as seemingly simple as growing cells in a dish could have such a profound impact on global health.
Yeah.
And you mentioned other applications beyond medicine. What else are cell cultures used for?
They're incredibly versatile. Think about cosmetics. Many companies are now using cell cultures to test the safety and effectiveness of new ingredients, replacing animal testing in many cases.
Oh, that's great. That's a win-win for science and animal welfare. What about other areas like, environmental science?
Well, cell cultures, they can be used to study the impact of pollution on living organisms.
Oh, okay.
By exposing cells to different pollutants, researchers can assess their toxicity and their overall effects on cellular function.
So it helps us understand the environmental consequences of human activities
Exactly.
and develop strategies to protect both human health and the planet. So those tiny cells are helping us understand the big, complex challenges facing our world.
Exactly. They're like tiny sentinels providing insights into the health of our planet and ourselves.
It's incredible to think about the vast applications of this technology, from developing life-saving drugs to creating a more sustainable future. It makes me wonder, are there any areas where cell culturing could have an even greater impact in the future? What's on the horizon for this field?
That's a great question, and the possibilities really are limitless. One area that holds immense promise is personalized medicine.
Okay, personalized medicine, I've heard that term before, but refresh my memory. What exactly does it mean?
So personalized medicine is all about tailoring medical treatments to an individual's unique genetic makeup and lifestyle.
Okay.
It's about moving away from that one-size-fits-all approach to healthcare and developing therapies that are specifically designed for you.
So it's like having a custom-made suit, but for your health.
Exactly. And cell culturing is playing a key role in making this vision a reality.
Okay.
Researchers are using cell cultures to grow and study cells from individual patients,
Oh, wow.
allowing them to test different drugs and therapies and see how those cells respond.
That's amazing. So instead of just using a standard cell line, they can actually use cells from the patient who needs treatment.
Precisely. It allows for a level of precision and personalization that was unimaginable just a few decades ago. Imagine being able to test different chemotherapy drugs on a sample of your own cancer cells to see which one is most effective for you.
Wow, that's personalized medicine at its finest. It's like taking all the guesswork out of treatment and tailoring it to the individual's needs. Are there any other exciting developments on the horizon for cell culturing?
Another area that's generating a lot of buzz is 3D cell culturing.
3D cell culturing. What's that all about? I thought we were already growing cells in the 3 dimensions in those petri dishes.
You're right. Traditional cell cultures are typically grown in these flat, two-dimensional layers. While that's been incredibly useful, it doesn't fully capture the complexity of how cells interact and behave in the human body.
So it's like comparing a flat map to a globe. They both provide information, but the globe gives you a much more realistic picture of the world.
Exactly. 3D cell cultures allow researchers to grow cells in more realistic three-dimensional structures,
Okay.
mimicking the way they organize and interact in tissues and organs.
That sounds like a game changer. How are these 3D cultures created? Do we need, like, tiny little scaffolds for those cells to grow on?
There are different techniques, but one common approach is to use biocompatible materials,
Okay.
like hydrogels or sponges, to create a 3D scaffold that cells can attach to and grow on.
So it's like giving those cells a tiny, custom-built jungle gym to climb on.
That's a great analogy.
I like that.
And these 3D structures, they're much more than just a cool visual.
Okay.
They allow cells to behave more naturally, form in complex networks, and interact with each other in ways that resemble what happens in the body.
Okay, so what's the big deal with 3D cell culturing? Why are scientists so excited about it?
Well, for starters, it allows for more accurate drug testing. Remember how we talked about the limitations of those traditional 2D cultures? Well, 3D cultures, they provide a more realistic environment to test drugs. Giving us better insights into how they might behave in the human body, this could lead to the development of safer and more effective drugs with fewer side effects.
So it's like upgrading from a basic flight simulator
Yeah.
to one that actually feels like you're flying a plane?
Exactly. And it's not just about drug tests and 3D cell cultures, they're also being used to study how tissues develop and repair, paving the way for regenerative medicine and tissue engineering.
Okay, now you're really blowing my mind. Are we talking about growing organs in a lab? Is that even possible?
It's definitely an area of active research. Scientists are already using 3D cell culturing to create mini organs, also known as organoids,
Okay.
that mimic the structure and function of real organs.
Wow, that's incredible. What kind of organs are we talking about? Hearts, lungs,
Yeah.
brains?
Researchers are working on a wide range of organoids,
Wow.
including those that mimic the liver, kidneys, intestines, and even the brain. These mini organs, they provide valuable insights into how organs develop, function, and respond to disease.
So we could potentially use these organoids to study diseases in a dish, test new drugs, and even develop personalized therapies.
Exactly. The possibilities are truly remarkable.
This whole field of cell culturing is just mind-blowing. It's amazing to think about the progress that's been made and the incredible potential that lies ahead. But before we get carried away with all the amazing scientific advancements, let's not forget that with great power comes great responsibility, right?
You're absolutely right. The ethical implications of cell culturing are something we need to address head on. As we delve deeper into manipulating living cells, especially human cells, we need to have open and honest conversations about the boundaries of scientific inquiry and the potential consequences of our actions.
Okay, so let's talk ethics. What are some of the key concerns surrounding cell culturing?
One of the biggest concerns is the source of the cells themselves. Remember those HEK293 cells we talked about earlier?
Yeah.
They're derived from human embryonic kidney cells.
Right.
The use of embryonic cells, even cell lines that have been around for decades, raises questions about the origin of those cells and whether their use is ethically justified.
So it's a question of where those cells came from and whether we have the right to use them for research.
Exactly. And it's not just about embryonic cells. The use of any human cells in research raises questions about informed consent. Were the individuals from whom those cells were originally derived properly informed about how their cells would be used? Did they give their consent? These are complex issues with no easy answers.
It's a reminder that even in the lab, we need to consider the human element, the origins of these cells, and the potential impact of our research.
Absolutely. And it's not just about where the cells come from, it's also about what we do with them. The ability to manipulate cells raises questions about the limits of what we should be doing. Should we be altering the genetic makeup of cells in ways that could be passed down to future generations? Should we be creating organisms with entirely new capabilities?
Whoa, that's venturing into some pretty deep philosophical territory. It sounds like we're talking about the very definition of what it means to be human.
You're right. These are big questions.
Yeah.
And they don't have easy answers. We need to have open and honest conversations about the ethical implications of cell culturing involving scientists, ethicists, policymakers, and the public.
It's a reminder that science doesn't exist in a vacuum. It has social and ethical implications that we need to address head on.
Exactly. And ultimately, it's up to all of us to decide how this powerful technology should be used.
Wow, we've covered a lot of ground in this deep dive. From the technical intricacies of working with cells to the incredible potential of 3D cell cultures and personalized medicine, it's been quite a journey. I feel like I've earned my PhD in cell culturing. But before we wrap up entirely, there's one more question I've been pondering.
I'm all ears.
We've talked about how scientists use cell cultures, but can anyone explore this fascinating world? I mean, I don't have a lab in my basement yet, but is there a way for the average person to, learn more about cell culturing?
Absolutely. There are so many ways to delve deeper into this field, even if you don't have access to a lab.
Okay, good.
You can start by reading books and articles about cell culturing or watching documentaries about scientific breakthroughs made possible by this technology. Many science museums, they also have fascinating exhibits on cell biology, bringing those tiny cells to life in a way that's both informative and engaging.
So it's about sparking that curiosity and seeking out information.
Exactly. The more you learn about cell culturing, the more you'll appreciate its impact on our lives and the incredible potential it holds for the future. Who knows? Maybe some of our listeners will be inspired to pursue careers in science or research after learning about this amazing world.
I love that. We need more curious minds pushing the boundaries of knowledge and using science to make a positive impact on the world. Now, as we wrap up this deep dive, I want to leave our listeners with one final thought-provoking question to ponder.
You're away.
What role do you believe cell culturing should play in shaping the future? How can we harness its power to address global challenges while ensuring its responsible and ethical use? It's a big question, but I think it's one we all need to grapple with as this technology continues to evolve.
It's a question that deserves careful consideration and open discussion. And to our listeners, I hope this deep dive into cell culturing has sparked your curiosity and inspired you to explore this fascinating field further. Remember, the world is full of wonders, both big and small, waiting to be discovered.
Well said. And on that note, it's time to wrap up this episode of The Deep Dive. A huge thank you to our expert guide for leading us through this incredible world of tiny cells and big discoveries.
It's been my pleasure.
And to our listeners, thank you for diving in with us. We'll see you on our next adventure. Welcome back to The Deep Dive. It feels like we've traveled through this microscopic universe, exploring cell culturing. From those basic lab setups to the mind-boggling potential of 3D cell cultures and personalized medicine, it's been quite a journey.
It really has.
It is incredible how much we can learn from these tiny cells, isn't it? They hold the secret to human health, environmental sustainability, and even the future of food production.
I know, it's amazing.
It is amazing. But before we get too carried away with all the exciting possibilities, I think we need to circle back to something that we touched on earlier, which is the ethical considerations surrounding this really powerful technology. Because, it's all well and good to talk about scientific advancements, but we need to make sure we're using this knowledge responsibly.
Yeah, you're absolutely right. I mean, manipulating living cells, especially human cells, raises a whole host of ethical questions that we can't just brush aside.
So let's dive into those ethical considerations then.
Yeah.
What are some of the, key concerns that researchers and society as a whole need to be thinking about?
Well, one of the major areas of concern is the source of the cells themselves.
Sure.
Remember those HEK 293 cells we talked about earlier?
Yeah.
Which are derived from human embryonic kidney cells. The use of embryonic cells, even cell lines that have been around for decades, raises questions about the origin of those cells and whether or not their use is ethically justifiable.
So it's a question of where those cells came from and whether we have the right to use them for research, even if it could potentially save lives.
Exactly. And it's not just about embryonic cells either.
Right.
The use of any human cells in research raises these questions about informed consent. Were the individuals from whom those cells were originally derived properly informed about how their cells would be used? Did they give their consent? These are not easy questions to answer.
Yeah. It's a good reminder that even in the lab, we can't just detach ourselves from the human side of things. You know what I mean? These cells came from somewhere. We need to respect that.
Absolutely. And it's not just about where the cells come from. It's also about what we do with them. The ability to manipulate cells, to alter their genes, to create entirely new cell lines raises a whole new set of ethical dilemmas.
Okay, I'm sensing we're about to wade into some deep philosophical waters here. What are some of those dilemmas?
Well, think about it. Should we be altering the genetic makeup of cells in ways that could be passed down to future generations? Should we be creating organisms with entirely new capabilities? Where do we draw the line between scientific progress and responsible stewardship of life? These are questions that society as a whole needs to grapple with.
It's almost like we're writing the rules for a whole new world, a world where we have the power to shape life at its most fundamental level.
Yeah.
That's both incredibly exciting and a little bit daunting.
It is, and it underscores the importance of having those open and honest conversations about the ethical implications of cell culturing. It's not just a matter for scientists to decide, it's something that needs to involve ethicists, policymakers, and the public as well.
It's like we need like a global town hall meeting to discuss the future of cell culturing and make sure we're all on the same page.
That's a great way to put it. We need to ensure that this powerful technology is used for good, that it benefits humanity and the planet, and that it doesn't lead us down a path we might regret.
Well said. This has been an incredible deep dive, filled with both like mind-blowing scientific advancements and thought-provoking ethical questions. I feel like I've learned so much about this really intricate world of cell culturing, the techniques, the potential, the challenges, and the responsibilities that come with it.
Yeah, me too.
And to our listeners, we really encourage you to continue exploring this really fascinating world. Read books, watch documentaries, visit science museums. There's a wealth of information out there just waiting to be discovered.
That's right.
Yeah.
And remember, the world is full of wonders, both big and small.
Well said. On that note, it's time to wrap up this episode of The Deep Dive.
Yeah.
A huge thank you to our expert guide for sharing your knowledge and insights with us.
It's been my pleasure.
And to our listeners, thank you for diving in with us. We'll see you on our next adventure.